I'd like to introduce our speakers this afternoon. Walid EI Azab, Senior Technical Service Manager for STERIS Corporation. He's very active in the organization for ISPE, PDA, ECA, and other numerous organizations. He's published various articles, and he's a QP and a Sigma Green Belt. We also have Richard Chai, who is also Technical Service Manager based in Singapore. He is a common face at our events, having spoken frequently for us, for which we're grateful. And so I will now hand over to them.
Thank you, Linda. Thank you. Hello, everyone. And like I read in the chat, hello, world. So I hope you are seeing my screen.
Yes.
That's perfect. Let me do one last thing. Here we go. Oh, it's excellent. So as suggested, we will go through the draft Annex 1 on cleaning disinfection requirement. But again, it's not just about the draft Annex 1. This is a title to make it a little bit exciting, but it's also about all type of regulation. So really the goal is to see beyond regulatory compliance and making sure that we have an adequate cleaning and disinfection process that is capable to control our bioburden in our clean rooms. So the scope of this presentation will be only on non-product contact surfaces. That's mean clean room surfaces like the floors, walls, ceilings, etc. So during this presentation, we will go through the regulatory guideline and making sure very quickly, making sure we are all on the same page, and look on the different cleaning and disinfection program, the design, validation, implementation, and Richard Shai will present an excellent case study that has been published, and then we will look at the program evaluation. Again, it is a life cycle approach, like anything in our industry. More and more, we look at our processes as a life cycle approach. So from a design to a validation, implementation, evaluation, and an improvement to making sure that we always improve and making sure we have an adequate contamination control program or strategy. So first of all, just to be clear on the definition, when we say sanitation, this means a removal of organic and other type of residue to reduce to a certain level the contamination. It does not provide any full kill or any full removal. There is also the term cleaning. We all know what it means, cleaning, but it's more about removal from a facility, an equipment, or a surfaces of a product, of a chemical or organic residue. In this case, we are looking at a cleaning of non-product contact surfaces and not the cleaning of process equipment such as a vessel or a filling line, etc. Then we have the term decontamination. Really, the term decontamination, some may even say biodecontamination, but the goal of the decontamination is, again, reduce to a certain level an unwanted matter that is normally hazardous. Either it's biological or it's a chemical compound. And we say bio because it's for, organic compounds that you try to remove, okay? So then we have sanitization, and this, again, it's about more reducing an inanimate surface, but in that case, it's just on all type of living form, like microorganisms, fungi, and yeast. Disinfection. The disinfection is a more regulated term in the fact that you have to demonstrate a certain kill on an inanimate surface, for example, stainless steel, epoxy, based on lab studies through a suspension or a coupon studies. And our last term that may be used is sterilization. This is well harmonized through the different pharmacopoeia, so I don't need to go into the details of what does it mean. If we look at the different regulation, either the draft Annex 1 or the current Annex 1, the PICS Annex 1, the Japanese guideline on sterile manufacturer, for sterile manufacture, the US FDA guidelines for aseptic manufacturing, and the CFR, the different wording that is used is is similar, and the processes in a way to implement a cleaning and disinfection process seems to be similar. The one that give a lots of details on the way to do it from my perspective is a Japanese guideline, where it's really explaining how to implement it and what to do if there is some, failure or some challenges. There is also USP guidelines and etc. that go into the details. So for example, the 1072 that go into the detail and give also adequate and good example, for example, on the log reduction, but also on the type of disinfecting agent that can be used. And finally, the WHO. We will be looking through the draft Annex 1, the version 12, on the different information given into the chapter of the disinfection around the design, first of all, around how to map your process and how to map your clean rooms to be able to identify the adequate selections of your disinfectant based on a cleaning and disinfection risk assessment, and we will look at through this. And then also how to validate your disinfecting agent, how to design your procedure. And again, Richard will present a case study. And through your historical environmental monitoring data, see how this cleaning and disinfection procedure can be improved. So the first step is really about mapping the process. And the goal of mapping your manufacturing process is first, to understand the different steps of your processes. Second thing is to understand the source of contamination. Really, the goal is about that. And again, you need to identify what type of source, what type of contaminant are of your interest. So in our case, it will be more micro and removal of residue if there is a residue on the surface. So for example, in an API manufacturer, the goal will be first to remove the residue and then disinfect using a disinfectant or a sporicide. So we will look at all that. So the type of process will also help you in understanding where are the source of contamination, but also the probability of having a contamination, for example, versus a dry process versus a wet process, closed process versus an open process, non-sterile versus sterile. Then the location, where in the area, and we will see through some benchmark. So we perform some survey. And so you will see a little bit how the practice is. It's not a benchmark, it's more about the practice around the industry of how do they define, for example, the frequency of cleaning and disinfection, how they base this frequency, for example, and etc. But the area, the classification of the area is also important because it puts up for you the specification, the microbial specification of this area based on the Annex 1. And then, of course, is your surface area, or for example, your floor is contain a high residue, level of high residue. So that means you have lots of powder on your floor and etc. That may alter the efficacy of the disinfecting agent. Then there may be a need to first clean and then disinfect. This we will also discuss, and when to apply a two-process cleaning first, then disinfection, or having a one process cleaning and disinfection in parallel. After you have mapped your process, it's also important to map the clean room. And in the clean room, you want to look at the clean room environment, for example, the temperature, air renewal. Why? Because it will have an effect on your wet contact time, which is very important, but also an effect on what type of technology you will be using. The clean room activity, the flows, the activity, shift numbers, material, number of person in the clean room will have an impact on your risk assessment and will have therefore an impact on the type of disinfecting agent or even the frequency to be used. The clean room surfaces, so the material of construction, compatibility, accessibility, if there is surface condition, all that needs to be taken into account when you are defining. And then of course, your air or your surface quality based on historical EM data, which will drive the frequency, but also the type of disinfecting agent to be used. When I say disinfecting agent, that means disinfectant and or a sporicide. When I say disinfectant, that means a broad spectrum disinfectant that is only capable to kill vegetative microorganisms. Sporicide, we understand, is capable to kill all type of microorganisms and spore former microorganisms. The source of contamination in your clean room are generally coming from generally. All the time coming from people, the transport of the material from non-classified or from a lower grade to another to a higher grade, the treatment of your surfaces due to the residue from your detergent, your disinfecting agent, from the personnel, from the equipment, etc. The drug component, so if you have spillage, if you have open processes, and then of course the process equipment may release some particles in some occasions. Again, when you are mapping your process also, the goal is to understand what type of surface do you need to clean. So here the goal is really, again, to list the surface and make sure you have all the different surfaces that needs to be cleaned. And then based on that is the application technique. A vaporization of a fumigation is generally used for large volume, closed system like isolator, wraps, material airlock, etc. Or area that is very difficult to reach by using a mopping or a wiping traditional approach. Generally, if you are using a mopping or a wiping, it's for large surfaces, smooth surfaces, etc. A spray is generally performed on small material, for example, or a transfer of material from a grade to another grade. Again, the way to spray is also important to make sure you are covering the different surfaces, but also you want to make sure you're not, you're not a happy trigger spray person where you spray a lot and then you have itchiness issues. So why understanding your process is important? Because it will allow you to be able to develop an adequate risk assessment and a robust risk assessment that is helping you in defining. And I use this from the BPOG for environmental monitoring risk assessment that they have developed. It's a great document, and I think for those who are doing risk assessment is I think is the best to read. That's my thinking again. So here what they did is they look at they put the different steps. You all know how to do a risk assessment, so I don't need to go into the detail, but they put the different steps, and then here the variable that may lead to contaminations. And then they score it. The advantage of that, it's help you to perform a heat map where you can define where there is a high risk of contamination, and therefore a need to put some controls and a need to put procedure to remediate to this risk and reduce your residual risk. You may also add it, plus those that is here, the activity length, the number of shifts, the presence of contaminant that may alter the disinfection. So if, for example, if you have a lot of residue on your surfaces and the disinfectant does not have an adequate blend of surfactant and it's not capable to clean, like for example, alcohol or a sporicide, then there may be a need to have a first cleaning step before disinfecting the surface. So again, to summarize, really map your process, identify the source, evaluate the contamination risk, and perform your risk assessment. This will be a great input to define also the frequency of disinfection, the type of disinfectant to be used, and especially the technology to be used. So really leverage the EM risk assessment to help you define the adequate frequency and the technology and the type of disinfecting agent. Of course, this is applicable for a new facility. If you have a facility that is already existing, another parameter that should be used is the historical environmental monitoring to help in defining the type of disinfecting agent to use and the frequency. Now, there is a lots of discussion, again, around disinfectant rotation. So that's mean using a broad spectrum disinfectant and rotate to. And so if we look at the different guidelines, so here is from Europe, MEA. They say more than one time, it's not the draft. More than one type it's the draft. The more than one type disinfecting agent should be employed to ensure that where they have different mode of action and they combine is effective against all my bacteria and fungi. Disinfection should be included in a periodic use of a sporicidal agent. Again, they use the term disinfecting agent and they have to be effective against all bacteria and fungi. When we look at the US FDA aseptic guideline, it proposes or it suggests the use of a routine disinfectant, so that means capable of killing a gram-positive microorganism, again, the vegetative floor, and to have a rotation with a sporicidal agent. However, if your historical EM data based on associated adverse trend, deviation, recurrent deviation, and etc., shows that, for example, there is a sensibility for a microorganism to pop up all the time, then there may be a need to have another type of schema, for example, having 2 disinfectant with a sporicide, okay? The goal of having 2 disinfectant with a sporicide is to be capable to achieve and kill the larger, more microorganisms and to cover of a maximum the microorganism spectrum. So if you are using 2 disinfectant of a different brand, but that have the same profile of killing microorganisms, this is no more rotation. Okay, so really you need to look at that, and you need to look that they have different spectrum of killing to be able to call it a rotation of disinfectant, for example, based on your environmental monitoring data. And the Japanese guidelines tell it well, or suggested well. They say if a disinfectant might have an inferior efficacy against a microorganism isolated from the environmental, then a re-evaluation, a replacement, or an alternative use of different disinfectant should be considered and implemented as appropriate. And of course, if you have spore former into your EM monitoring data, then there is a need to use a sporicide agent. Then, of course, you have also national laws, which says that you have to use 2 disinfectant with a sporicide. You don't have a choice. Here it's a national law. We have to apply the national law. In that case, we don't have a choice. If we look at the industry association, the USP is really effective on that. So 1 disinfectant with a sporicide is enough. However, there is other rotation scheme that maybe support as we just discussed. And the PDA go also in the same line of this one. So in conclusion, it is acceptable to have 1 disinfectant and a sporicide if it is supported through a microbial risk assessment, and also if your historical EM data show that you have adequate control of your bioburden. If you start to see pop-up of microorganisms that cannot be controlled with the type of disinfecting agent that you have, then the need to have an alternative or a replacement is to be looked at. So here we did a benchmark, a pool survey, sorry, not a benchmark, a pool survey to understand the practice around the industry. So here in blue is the people who answer from where they are from. And so we asked them if they are rotating more than 1 disinfectant with a sporicide. So that means 2 disinfectant with a sporicide. And 55% say yes, 45% say no. And when we ask them why, for those who say yes, why you are rotating more than 1 type of disinfectant, you can see that, so here the 54% is not applicable because of this, but because of the no here. But the majority is based on historical EM data. Some says it's because they want to be compliant with the MEA and the PICS guideline, and 9% says that they have to be compliant with the national regulations. Okay. We had more than 100 people answering those pools, and so I guess you give a good idea of what is the practice around the world, if I can say that. Now that we have looked at, do I need to rotate 2 or 1 disinfectant? Now let's look at another point that is well discussed in the industry is, do I need to clean prior to disinfection? So that means, do I need to have a separated cleaning using water, detergent, whatever you are using as a cleaning agent, rinse it, and then disinfect? It's a good practice after a detergent to rinse it, because the detergent has a lot of surfactant, which can be a lot of residue that may interact with your disinfectant, except if you can demonstrate the contrary. So it's a good practice to rinse between. So the goal is to see, do I always need to clean with a detergent, rinse, and disinfect with a disinfectant or a sporicide? So to be able to do that, let's try to understand what are the parameters or the element that may interact with your cleaning or with your disinfectant. So first of all, we have seen it, the residue that can interact. The other point is also the concentration or the composition of the disinfectant. Does it contain a blend of effective surfactant, or is it APA registered as a cleaner and a disinfectant? APA is from the US APA Environmental Protection Administration, I think, or as a citizen. The third part to look at it is the actions that it's put. Are you spraying or fumigate or vaporize? So if I'm spraying or fumigate or vaporize, even if my disinfectant has some capability to clean, there is no mechanical action, so it will not help in cleaning the surfaces. Mopping and wiping will help to have a mechanical and also help to clean and disinfect. Another point to look at it is the environment. So a cold room, the temperature is very low, so that means the kinetic of the killing will be very low. So you want to make sure either to keep the wet contact time adequate or maybe to have a cleaning prior. So what we did with some of the industry folks is we tried to develop I would not call it a risk-based, but more a number assessment. Do I need and to clean prior to disinfect? So what we look is the different parameters that I have exposed to you. And then we look, the number one is mean it's very easy. The number three is high risk, or it's very difficult. So do we know if our residue have an interaction with the disinfectant? No. Okay. If yes, then it's a number three. If we don't have a data, we put ourselves between a two. Again, it's an example. If you want to use other numbers, it's up to you. Is the amount of residue on the surface visually visible? Yes is a 3, no is a 1, etc. And we go through it. And the goal is to help you ranking it. So if you multiply 1 by 1 by 1, it's a 1. If you multiply 2 for those, it's 32. And if you multiply the 3 by those, is a 244. Based on that, based on this assessment, from 1 to 12 is a no. You don't need to every time clean prior to the disinfection. And this should be the majority of the case for sterile manufacturer, biotech manufacturer, and etc. Or closed system manufacturer, even ITMP. However, if you are above the 12 or another number that you have defined for your processes, then there may be a need to first clean prior to a disinfection. So this gives a little bit of, we have looked at all the parameters to assess if there is a willingness. It puts some kind of justification behind your practices. Qualification of the wet contact time is very important. In the lab, the way you are performing it is you're using a coupon, you put a microorganism, a number of microorganisms on the coupon, and you look after a certain time if you have, a microbial kill. And generally, this would depend on the type of disinfectant used, the type of surface used, and the concentration used. So we will be looking at that to assess what is the wet contact time that should need to be used in your facility. Of your process. And in this phase 3, when you do it in the field, the goal is to look how it's different variable from the clean room. So the temperature, the humidity, the air exchange, the application technique, the surface cleanliness will affect the efficacy of your disinfecting agent. And if the wet contact time validated is enough for the field validation. It is important, therefore, to make sure that during your application on the field, in routine, that you are respecting the validated wet contact time. Okay, we have seen a lot of 483, and this is a recent 483 from 2016, recent 5 years, where it speaks about a manufacturer not respecting the wet contact time that it has been validated in vitro versus what is being done on the facility. So now is when to clean. So how may I define the frequency of the cleaning? Majority of manufacturers, and we will see through the survey, look at it as the grade area. So the first look is to look at the classification area, grade A, grade C, grade D, The more you are in the classification, the more you will be, or the more frequently you will be cleaning and using a higher type of disinfecting agent. Okay. The activity in the clean room is also important. So if the number of shifts, you are in a one shift and then you're starting to pop up into your, to ramp up into your activity, and you started to have 2-3 shifts, then it makes sense to review and revise your cleaning and disinfection procedure to adapt it based on the activity in the clean room. Also, the surface localization. So for example, if it is a surface that it nears a product, or it's a surface that it's an indirect product contact, and etc., or it's a surface that is highly touched, for example, or operator have a highly intervention frequency, this also need to be taken into account. And of course, always historical microbial data should be taken into account if you have those historical microbial data. So the goal after is to perform your ranking and then define your frequency. Of course, here will be the lowest frequency to the highest frequency. I didn't put the daily, monthly, and etc. because I think it's up to the manufacturer to decide what is the adequate frequency based on their activity. And after here, it's an example of how to put in a SOP. Make sure you put it through a table so it's easy for people to read rather than a long wording. You forget what you have read at the beginning of the page. So here, as a practice, 39% of the people that we surveyed say that they base the frequency of cleaning and disinfection based only on the clean room grade classification. We have only 37% that do it through a throughout microbial risk assessment. So that means they're looking to all the different parameters that, or elements that may affect the frequency, that may affect the frequency. That needs to, that all the parameters that needs to be taken into account to decide the adequate frequency. Other will look at historical review EM data and the number of excursions. So the more they have number of excursions, the more we increase the frequency. Again, you have excursion. By increasing the frequency is a very short-term remediation, just to make sure you are controlling your environment, but you need to find the root cause. Otherwise, it will stay for long. And you still have 5% that rely on the supplier recommendation. It's okay. Again, as a manufacturing authorization holder, you are the only one responsible to ensuring that you're controlling the bioburden of your clean room, and therefore the product quality and safety to the patient. After you have defined all that, so what type of disinfecting, rotation or not, clean before, frequency, etc. Now we need to go on the floor and train the people and train the operator, transfer this knowledge to them. So either you are using 3 bucket, 1 bucket, 3 bucket, 2 bucket, is up to you. Some may use 1 bucket because they have single-use mop. That's also acceptable. Again, depending on the type of mop you are using, depending on is it a single or non-single use, all that we will be looking at. The dilution should be performed. If you have a grade D and a grade C, should be performed using the water quality of a purified water. And if you are in an aseptic area, like a grade A or a grade B, should be with water for injection. That's the minimum. Some expert will say it is preferable to use only water for injection because it has a lower bioburden charge. But I think Wi-Fi and purified water for the different grade is enough. It can be done through a graduate cylinder, a pump system, a pre-measure unit dose pack, or a ready to use, but you want to make sure that your operator is adequately trained to perform that. Then also, after the dilution, the goal is also to assess what is the change out use dilution. So that would mean after how many meters square I need to replace the disinfecting solution that is in my bucket. And for that, you do have different types. So here, the practice in the industry is either a visual inspection of the surface of the mop or of the disinfecting solution. Okay. Again, if it is a visual inspection, that means you need to train your operator just to make sure that someone that say the mop is dirty, the other one say no, it's clean. You don't want to have some variability in that. So there is a need of a little of training. Some based it on a predefined area. So for example, they would say every 20 meters square or for one room, you use one bucket with one disinfecting solution, and after a certain time, you change it. Other will divide the room into a different grid based on historical observation, based on visualization of the clean room clean status, based also on a wet contact time, how long does it stay wet, for example, and also on the substrate concentration. So if we look at the practice, we have 41% that generally define the change out of their bucket based on the supplier recommendation. So I really hope that in that case, the supplier went on the floor, look and visualize the process to be able to say after how many meters square, for example, you have to change your bucket with a new solution. Others, 24%, look at a defined area, that is, they have defined a specific area, or for example, 20 meters square, to be clean and disinfect per bucket. Other use Anne-Marie Dixon rules, which is every 600 meters square for an ISO 5 and a 6, every 1,000 square foot, every for an ISO 7 and 8. And 16% based it on visual inspection of the solution. Now that you have spoken about the solution, now let's spoke about the mop and the wipe to be after how many time I can remove. So depending on if it is a single use or not a single use. If it is a single use, you want to make sure that you look also with the supplier of how many meters square it is available, but then it is capable to cover. But then you will have to look at the visual inspection and the mop cleanliness and also the mop shelf life. If it is a reusable mop, you want to make sure the number of cleaning cycle is adequate to not alter the material of construction of the mop. So if your mop arrive when it is a new mop, a blue mop, if your mop start to change color, that means it's maybe time to change the mop. Or you define it based on a predefined area. We look at the practice, 48% change their mop or wipe based on a visual inspection performed by the operator. I hope they are well trained. 22%, again, based on the supplier recommendation. And then we have 18% that have performed microbial analysis and plus visual inspection of the mop to demonstrate that the mop is still adequate to clean and disinfect and remove, and after cleaning and after the reuse, the micro on the mop is okay. Okay, and we still have 11%. I put this sensor. I didn't imagine that I would have answer on it, but okay, I put the sensor and I had when the mop change color and look destroyed. This is too late. The way to wipe, either you have ready to use wipe, and in that case, you can take the wipe and really wipe on the surface. If you don't have a ready to use wipe, you may need to pre-impregnate, you may need to spray on the wipe and making sure the wipe is enough wet. And the goal of enough wet is that the wipe is enough wet to, when you are passing through a surface, it allows to release the water or to release the disinfecting agent on the surface and allow a certain film to stay on the surface to be wet and to achieve the kill. If you're not wetting adequately the mop, which I see a lot, the mop is not wetted adequately. And what happens is the operators start to mop the surfaces and the surfaces is not even wet, or it's wet, but not enough to stay the adequate wet contact time. So that makes sure to have that. Some people may even take a bunch of mop, put it in a bag, put the disinfecting agent, and then shake it. Okay, I want to make sure this is done adequately. I'm not I'm not in favor of that. That can be done. So if we look at the way people is doing, 48%. No, sorry. Here we go. I will not go through the way to clean and disinfect, but I think we all know now that you have to go from the most critical area to the least critical area. So that means from the most cleaner area to the most dirtiest area. So that means at the end of the room to the towards the door, if I'm looking at the wall, from the ceiling to the down or to the floor to clean. It has to be done with an overlapping of by 10-20%. Either you are using a stroke or either you are using a S shape. Whatever you are using, make sure it is applicable and your operator is adequately trained for this method. So now that we have seen how to apply and how to disinfect our surfaces, so after we have disinfected our surfaces, we need to remove the residue. Residue, like I said, come from disinfecting agent, come from the people, come from the product, come from the material, and etc. So it's really a blend of residue that may alter your disinfecting agent, okay? We have data, and we have enough data to demonstrate that just residue of the disinfecting agent will never alter the disinfection process if the two disinfecting agents, of course, are compatible with each other. So what should be looked and what are the parameters to look to define your frequency of removal of disinfecting agent? The first thing is the appearance of residue. So that's mean when it's visually visible that the residue is there. Material of construction is also important. So you will look that for transparent surfaces, residue will be more quickly visible than for a stainless steel or an epoxy or a white Lexan, for example, where there would be more time before you can see the residue. The interaction of the residue with the disinfecting agent being used, that's also another parameter to look at it. Again, we did the same way with some numbers. So look at the appearance, material of construction, compatibility, frequency, interaction, and area classification. Why area classification? Because for a grade A and B, you would need, there is a most frequent use of disinfecting agent and etc. So there is a more need of a frequent cleaning and disinfection of a rinse principle. So this is why the area classification is introduced here. We look at that, and the goal is to say that the minimum will be based on the visual inspection. So more than 4 weeks, for example, this will be the minimum. And for example, if the appearance of the residue is less than a week, we put here between weekly and daily. Why weekly and daily? Because you would need to assess based on all that if I need to perform it daily. The things that needs to be looked principally is the material compatibility. If a material is known with a poor compatibility with the disinfecting agent that you are using, in that case, it's a 3, you may define a more frequent rinsing program. Based on your cleaning and disinfection program, if we are looking at a first option, so that's mean where my surface does not contain any residue, there is residue that is built up from the processes and etc. What happens is I am cleaning and disinfecting the surface, and at a periodic frequency, I'm performing a removal of those residue using water or IPA. In aseptic area, the water will be Wi-Fi, and in a non-aseptic area like a grade C or a grade D or a control classified area, purified water can be acceptable. And then we go back to the non-residue. If you are performing in the option 2, so that's mean I'm performing a cleaning prior to disinfection. In that case, no residue, I disinfect the surface. There is a residue on the surface from the disinfecting agent. Then I start to, I have residue from the disinfecting agent and the process. Then I have a cleaning process. I rinse the surfaces, except if you have justified that it's not needed. You don't have any residue on the surface, and then you start to re-disinfect. So again, it's really important that it's look like a life cycle. It's really a life cycle approach, and you need to assess which of those program, option 1 and option 2, is adequate per your process. The way to remove residue from the surface should be done the same way as you are disinfecting. So from the most critical area to the less critical area, using the stroke and using water. The goal is to use water. Purified water, water for injection, or IPA. Some may use hydrogen peroxide. That is also a good chemical to remove residue of disinfecting agent. So now if we look at the practices, 70% of the people who answer, of the pool who answer say that they have a residue removal program, while we have 30% who say no. 56% say it is based on the visual inspection of the surface. And other says that it's based on other type of analytical methods, such as TOC or other type of non-specific analytical method. It's 35%, and generally those is because they are high potent product manufacturer, let's say. And you have 10% that is relying the frequency of the residue removal based on the supplier.
Thank you, Walid. So next I will go through a case study on the cleanroom startup. So before I get into the case studies, just do a quick recap on the disinfectant validation components. So basically the disinfectant validation consists of two components. The first one is the in vitro test. So the in vitro test is basically the test conducted in a lab. It consists of suspension tests and also a coupon test. So the suspension test is not compulsory. Basically, the main objective is to use as a screening tool to see which disinfectant is efficacious against certain microorganisms. So this is basically to check the efficacy of various disinfectants. It does not take into account the substrates of the cleanroom. So the tests on the coupon, basically the coupon test is more representative in the sense that it involves the use of the cleanroom substrates and also the environmental isolates as far as possible in the testing process. So this will basically simulate the actual cleanroom conditions as close as possible. So this is a more representative test to see the efficacy of the disinfectants. So after the end user pass the coupon test, then they can start to use the disinfectants in the actual clean room. And this will basically go into the in-situ test. So in the in-situ test, I want the previous slide. So in the in-situ test, basically this can involve the use of environmental monitoring data during the worst case events to demonstrate the effectiveness of the products and the application methods in the real world. So these the worst case events can involve things like the cleanroom startup or after the production activities, and even the microbial excursion events in the cleanroom. Next. So the in-situ testing, so these are the guidance documents that mention about the in-situ testing. So if you look at the USP 1072, it mentioned the comparison of the frequency of the isolation and the numbers of microorganisms isolated prior to and after the implementation of a new disinfectants. And if you look at the FDA guidance documents on the aseptic processing, it also mentioned about the effectiveness of the sanitizing, obtaining the samples before and after sanitization. And if you look at the PDA Technical Report 70, it mentioned qualification of a new antimicrobial chemical agent should include both the lab, which is the in vitro test, and also the in-situ testing. Next. So next we look at the some of the reasons for the cleanroom shutdown. So typically cleanroom shutdown can be divided into two main reasons. One is the planned cleanroom shutdown, the other one is the unplanned. So under the planned shutdown, you have the maintenance, construction activities or renovation activities to your cleanroom, or you have the equipment repair or installation in your area, or if there is extended the holidays and you want to shut down the AHU to save energy, or if there's no production activities, you will want to shut down the AHU. So these are all considered as the planned cleanroom shutdown. Of course, if you have the unplanned shutdown, it can be due to the power outage or the AHU breakdown. So in this case, this kind of unplanned shutdown may affect your the quality of the products if there are production activities going on. Next. So next, the restoration activities of the clean room depends on the length of the shutdown. So some shutdown can be less than 24 hours, very short, and some can be weeks or months. And the type of activities during shutdown can also affect what are the restoration activities that you need to implement to restore the clean room back to the operational state. So this will include how extensive is your work during the shutdown. For example, I mean, for extensive renovation work, if the surfaces and the clean room are heavily soiled, then you may need to have a pre-cleaning step to remove the heavily soiled surfaces. And also for non-use or extended holiday, those surfaces and the clean room can be relatively clean. So it should be easier to restore back to the operational state. Next. So these are the main activities for the restoration of the clean room. So the first one is the initial gross cleaning. So this is basically to remove the heavily soiled surfaces, and then followed by the cleaning and disinfection strategy. So this, you will need to have a plan of what chemical agent, what disinfectants, process agent to use, and the sequence and the number of times of each of these to be used. So you will need to have a strategy to perform the cleaning and disinfection. And then the last one, of course, you need to have environmental monitoring plan as well, at which time point to conduct the environmental monitoring. I'll go through this later on. Next. So this is the typical restoration activities. So initial gross cleaning basically is to remove the soil and other contaminants. So for heavily soiled surfaces, I mean, you need to use a broom to clean it, or vacuum, HEPA filter vacuum as well in the grade A areas, or mop with water as well. So this is to remove the heavily soil surfaces before you get into the cleaning and disinfection stage. So of course, you will need to collect the EM baseline data. So check what is the bioburden load in your clean room prior to your cleaning and disinfection activities. So after that, you will perform the cleaning and disinfection using disinfectants and the sporicide agent on all the room and the fixture surfaces. So typically, you will need to use disinfectants and also the sporicide agent. So the sequence typically is to use a disinfectant first. And as Walid mentioned, that disinfectants typically has more surfactants, and as a result, it has a better cleanability compared to the sporicide agent. So typically, disinfectants are used first to clean and disinfect the surfaces, two steps in one, basically, to clean and disinfect the surfaces, and then followed by the sporicide agent to apply on all the surfaces to basically address those hard to kill microorganisms such as the bacillus or the bacterial spores. So this is typically the type of disinfectants used and the sequence. So one of the method is the so-called triple clean. So triple clean, as the name suggests, just like you clean 3 times. So in this triple clean scenario, typically you will just need to apply 2 disinfectants, 2 times of disinfectants, and then followed by lastly a sporicide agent. Of course, the number of times of the disinfectant use and also the sporicide agent use, there's not so much of a guideline to say you must use how many times. So the main objective is the end result. You must be able to meet the bioburden requirements for the respective clean rooms before you can turn it over to the production. So next. So next, I'll get into these case studies on the clean room startup. So this case study is basically in the University of Tennessee Health Science Center clean rooms in the US. So this is a university-owned contract manufacturing facilities. So in this case, the clean room shutdown is due to maintenance and some engineering work. So they need to shut down the AHU to perform some work in the clean rooms. And they employ a triple cleaning and disinfection strategy to restore the clean room back to the operational stage. So in this case, they use the first one is the low pH phenolic disinfectants, followed by a high pH phenolics disinfectants, and then lastly a sporicidal agent. Next. So this is a triple clean environmental monitoring plan. So as I mentioned before, the T0 is the EM baseline. So you want to have a baseline bioburden level. After that, apply a low pH phenolic disinfectants on all the surfaces and also the fixtures in the clean rooms. So after the application, you collect the EM monitoring data again. This is a T1 time point. So after that, apply a high pH phenolic disinfectants on all the surfaces again in the clean room, including the fixtures. So after that, you'll collect EM monitoring data under the T2 time point. So finally, after the application of the two disinfectants, they apply a sporicidal agent. So in this case, is the hydrogen peroxide and peracetic acid blend RTO is the ready-to-use version. So this is a sporicidal agent to basically address all the bacterial spores that is very challenging to kill. Yeah. So after that, T3 is the time point for the environmental monitoring after the application of the sporicidal agent. So next. So this is a ISO 5 clean room for filling areas. So these are just some of the examples of the sampling points. So red one is the air sampling, green one is the floor and the surface sampling, and the blue is the wall sampling. So this is a small filling areas. These are the sampling locations. Next. So this is a summary of the sampling results for the ISO 5 area. So you can see that T0 is the baseline. So for ISO 5 areas, so typically can be a very clean area. So in this case, you can see that even at the baseline T0, you can see from all the air sampling, surface sampling, and the floor sampling as well. Total counts is, I mean, the CV is only 6 CFU, and it is mostly coming from the floor. So as expected, floor is the most dirty part of any clean room. So you will see the counts mostly coming from the floor, and the rest are just very clean. So if you move, so T1 is the after application of the low pH phenolics disinfectant, and then the count reduced to 2. And then T2 is basically the EM data for the time point where after the application of the high pH phenolic disinfectants. So it reduced to 1. So finally, T3 is the time point for the EM data for after the application of the sporicidal agent. So you can see that all have no counts. So you can see that there's a trending of the microorganism downwards. Next. So this is a graphical depiction of the trend. So it's a very low count basically. So starting is 6, as you can see that the floor after the application of each time point, there's a downward trend. And in the end, it's all 0 counts for the ISO 5 clean rooms. So typically it's expected because ISO 5 clean room can be very clean area. So next. So next we look at the ISO 7 areas, which is the grade C areas. So in this case, as mentioned before, the red one is the air sampling, green is the floor and the surface sampling, and the blue is the wall samples. So this is the compounding room. They got the material transfer room as well, and the gowning room. So this will have more counts as expected because it's a grade C area and also high traffic in some of the areas. Next. So this is the some of the data collected for the ISO 7 areas. So as you can see that there's more counts at the baseline T0 Typically, it's from the floor. You can see the floor of this room. And then 12 and 26 count is, again, is from the compounding room, mainly from the floor. So after the application of the LPH, low phenolics disinfectants, the count reduced. Of course, except there are some outliers in the floor in this room. So in this case, there are some outliers in here, even after the application of the low pH and the high pH phenolic disinfectants. The rest shows a very good result, downward trend. Of course, after the application of the sporicidal agent, you can see that all the counts reduce further to a very low count, which can meet the bioburden requirements for these few rooms. Next. So these are the more rooms in the ISO 7 areas. As you can see, some counts at T0. So in this case, there are some outlier as well. So during the investigation, basically, it shows that there's some high traffic area, and also the due to the material transfer using the trolleys, so that causes the outlier points. So the rest are very good results. And you can see that after the application of the sporicidal agent, time point T3, so all the counts is reduced to 0 except the 1 count. So typically, it's examples of the cleanroom startup where there's a downward trend after the application of the disinfectants and also finally the sporicidal agent. So this can be a very good data to show the effectiveness of your cleaning agents and the sporicidal agent under the worst case conditions of the cleanroom startup. So over to you, Walid.
Thank you, Richard. And the mouse is not here. Okay, I don't know why the mouse is not working anymore, but so now we will look at the ISO 5 core. So that's been grade A, but also grade B area, and the cleaning of those material. The first things to look is anything that is entering into an aseptic area needs to be either sterile or adequately disinfected or disinfected if it cannot withstand a sterilization process. And therefore you may have, for example, if you know the stopper balls, for example, that is an indirect product contact surfaces, and in some occasion we are not able to remove it from the filling line or from the isolator. In that case, it's not possible to clean it and to sterilize it. Therefore, it needs to be performed manually, so you can clean and disinfect the surfaces using either alcohol or a detergent, rinse, and then using a sporicidal agent. Whatever you are using, it's important because it's an indirect surface contact, product contact surfaces. It will fall under the Annex 15 around the making sure that the residue of the disinfecting agent or the detergent that you are using is you have an acceptable carryover for the product, and therefore you would need to have the PDE value and calculate this maximum acceptable carryover, and to assess if your cleaning process or your cleaning and disinfection process is adequate. So here we look at the cleaning and disinfection procedure, and we ask if does your procedure require a separate cleaning step before a disinfection of an aseptic area? So that's mean a grade B area, a class 1000 area based on the ISO. And we can see that 44%, let me see if I can do that. Here we go. We can see that 44% say no. So that's mean they don't have a clean first, then disinfect. Yes is 26%. That say they clean with a detergent and as a cleaning agent, and then of course they rinse and then disinfect. Then you have 22% that say yes, we clean with a detergent and water is also used. We have 5% that use water, and then you have the rest that you're another type of disinfecting agent, like a vacuum, for example. So now let's look at how we can clean and disinfect or decontaminate an isolator. So in the majority of the case, this is interesting. The draft Annex 1 is putting this as a cleaning prior to the disinfection step is essential, especially if you are using a VHP system where there is no cleaning process in place. It's more about the disinfection. So you want to make sure that there is no gross contamination or residue that may alter the decontamination process using, for example, vaporized hydrogen peroxide. And so the goal here is to look at the way to do. So the first thing is to clean, either using a detergent, using water. Some may use IPA with a mechanical action, of course. And define the frequency of this cleaning. It can be either after a shutdown, or either after a maintenance, or at a periodic frequency, based on what you think is the most adequate. The spill management is a little bit different. So if you have a spillage of product, then in that case the goal is to contain the spillage, and not to cleaning directly, but to contain the spillage until you have finished your production process. And then what happens is you start to disinfect your isolator using either a VHP. Some may use also a sporicidal agent, but the goal is really to try to use a VHP when you're in an isolator and in a closed system, because you can really disinfect in a closed system and keep the area decontaminated until the start of the production in that case. For the wraps, it's exactly the same thing, but instead of using a VHP, it would be a use of a sporicidal agent. Some may rotate a disinfecting agent as a cleaner with a sporicide, other will use IPA, other will use water, other will use detergent. Again, the type of chemical agent that you will be using should depend on your processes and should depend on the residue that is on the surfaces. Okay. Yeah, I say others use water. They will be using water on a specific area, but not everywhere. Generally, we tend to avoid water into an isolator or wraps. Okay, and so the way to perform it is perform a pre-cleaning, a cleaning prior to the disinfection. So if a sporicide is there, you will define the frequency. Again, the way to define the frequency, like the previous slide that I show, is to define the element that may alter, that may impact the frequency, and then look at those elements and define what should be the adequate frequency, or having a visual inspection of the surfaces to assess if there is visible residue on the surfaces that may alter the disinfection process using a sporicidal agent or using a VHP. Plus, you have the usage of the wraps during the process and during the production. Then we start to clean, disinfect, use the wraps, and then at a certain moment, the same way for an isolator, is to start to think about the rinsing frequency. So if we look at the benchmark for an ISO 5, that's mean a grade, in that case, a grade A, a class 100, is we ask the same question. So does your procedure require separate cleaning before the disinfecting, before the disinfection of a wraps or an isolator or a cabinet or a hood? And we have 33% who say no. That's interesting. But then you have yes, they use as a cleaner a disinfecting agent, which is 26%. Then you have yes, they use a cleaner and then they use a sporicidal agent. Okay, so some use only into the grade A disinfecting agent. Okay, I really hope that they are able to demonstrate that in their EM they don't have any spore former inside the grade A. And then 14% say yes, they use a disinfectant along with a sporicide. So that means they will use a disinfectant plus a sporicide with Resin. So that what happens is they use more frequently the disinfectant, and every week, for example, or periodically, they use a sporicide. Again, this is based on the microbial risk and based on historical EM data, of course. So now that we close the parenthesis for isolator and for ISO 5, we have how to clean our clean rooms and how to clean our critical area like the wraps, isolator, hoods, or LAF, or unidirectional flow. Now the goal is to go through the evaluation and assess what we have designed as procedure and what we have defined as type of disinfecting agent, type of technology to use, type of procedure that is put in place. Is it working? And the way to perform that, again, is to look at your historical EM data. But again, before looking at your historical EM data, you want to make sure that the EM data that you have is adequate. What I mean by that is that there is no practice leading to a high recovery of microorganisms. So what happens is you may have surfaces, for example, this is an example of a photo that has been took, where you have some mold on the surfaces, or where you have here an open and through the non-classified area, or where you have a little bit of rouge that is staining, or here it's open to a non-classified area, or for example, you don't clean your wheel carts, for example, adequately. Again, some rouge, again, some rouge. Here, it's a case study. I didn't present it, but it's a case study where it shows that the person, in that case, from 1 to 3, the wet contact time validated was 10 minutes, but the real wet contact time was 2 minutes, for example. So all those type of practice will affect your cleaning, your EM data by giving, pop-ups potentially or excursion in your EM data. So you want to make sure first to look at that through periodic audit, housekeeping, whatever you call it in your facility. But the goal is to try to understand if there is any data that may alter my analysis of this. So you clean the data, let's say like that. And then you will look into your EM, of course, of high recovery or microbial adverse trend, okay? The goal is to look at microorganisms that is popping up all the time. So for example, an excursion with Bacillus cereus or Burkholderia cepacia, you have several excursions due to that. And then the goal is to look at did I have tested this isolate? If it is a no, then it may be good to incorporate it in a future disinfectant efficacy using a suspension test as promoted by the USP 1072. And what happens is you will look if the disinfectant is working against this microorganism. If that's the case, maybe you should relook and improve your procedure. If not, then you maybe look at added another disinfectant, like suggested by USP 1072, the draft Annex 1 and the Japanese guideline on aseptic manufacturing. And if that's the case, review the procedure, recheck after recheck the actions put after a certain time. So for example, after 6 months, confirm that what you have improving the procedure is working. If that's yes, perfect. That means it's working and you don't have any more of those type of isolate. If not, then you may need to go and restructure your suspension studies and either confirm the efficacy or either use a coupon studies in that case to confirm the efficacy versus the wet contact time that you are observing. So the goal really about EM trending is to look at it and to try to understand your sample and where are the location where you are above, and based on that, take actions to improve. The goal is not to take action to increase the frequency. The goal is to take action to be able to improve and prevent this to happen again. To be able to do that, again, ISP is proposing a great training on deviation and kappa. And I think this is where the principal root cause should be. For the practice, 40% say yes, they have increased the frequency of their cleaning and disinfection because of historical EM data and historical EM data review. That's okay. Some say no, they never had to increase for the past 4 years the frequency of the cleaning and disinfection data. So that would mean they had excellent EM data, or they don't review it. And 25% say yes, they had to increase the frequency because they had recurring microbial, or they had microbial excursion, and as a remediation, they had to increase their frequency. So as a conclusion is, I like to say, try to keep it simple, okay? When it starts to be complex, that means there is something wrong. But it's not It's not a topic that is very simple too. But the only way to be able to put it simple is by really first understanding your processes, identifying where the source of contamination is, understanding what should be the adequate remediation and what are the elements that may affect the efficacy of your cleaning and disinfection. Make sure you're involving your operator and the people who is performing those actions, and make sure you adequately transfer this knowledge to those persons. When you empower those persons, you get more back into a more control, clean rooms and bioburden, but also more robust and more reproducible processes that is performed by your operator. So I think the key success here for a cleaning and disinfection is really about the knowledge of your processes, knowledge of the source of contamination, and also the transfer of knowledge to your operator. By that, I would like to thank everyone first for your listening, patience with us, and if there is any question, please do not hesitate.
Thank you, Walid. Thank you, Richard. We have several questions already, and Catherine K asked a question which Neo has already answered with regards to coupon substrate surface material. We have a question from Jong Hyuk. In fact, we have two, which is regarding in-situ study. How can we demonstrate the statistical comparison between before and after?
Yeah, I mean, for in-situ comparison studies, I mean, there's no fixed methods. And one of the methods you can base on is, I mean, if you look at the USP 1116, they talk about the contamination recovery test, basically the rate. It means what is the frequency of non-zero recovery of your plates when you do all the samplings. So basically, that is one of the methods you can compare. Of course, there are also other methods which can involve a specific, let's say, for example, a specific room. You can basically see the data on the T0, T1, T2, compile the total CFU for the specific room, and also can calculate the CFU per plate at each time point for the specific room. So these are some of the methods that you can consider. Of course, there are no fixed guidance on which one you must use. As long as you can use it to demonstrate that there's a downward trend after each cleaning, then that should be fine.
Thank you, Richard. The next question was asked by Ahmad Fauzi Musa, which was, is in vitro test required by medical device facility, and how can you clean and disinfect AHU ducting? That has been answered by Neo in the chat. Also, Jong Hyuk, your question about how can expiration of disinfectant concentrate be also, that has also been answered. Michelle asks, when preparing the EM cleaning validation protocol, as you are essentially starting with a dirty clean room and are likely to get counts on your plates, do you only set acceptance criteria after the final clean EM?
Yeah, that's a great question. You want me to go ahead, Richard, or do you want to go ahead?
Yeah, go ahead.
Okay, I think that's a great question. So the goal, of course, for example, after a shutdown, you would have a lot of microorganisms. So I don't think the goal is to set a criteria for before the cleaning and disinfection. The goal is to look at the criteria after your cleaning and disinfection, and demonstrate that your cleaning and disinfection program is capable in reducing the bioburden or the level of contamination to below the specification or below to your criteria. And that's exactly was the case study of Richard, who showed that, for example, for certain shutdown, you may need to have only one disinfectant is not enough. You may need to apply another disinfection. So a triple clean or a two clean, whatever you call it. Some call it two clean, so they do 2 disinfection and then a sporicide, or 3 disinfection and then a sporicide. And when the shutdown is very complex, they would do 3 disinfectant, 2 sporicide, and even I have seen that. So that's also a possibility. But the goal is to demonstrate that your cleaning and disinfection program is capable to reach your specification, your micro specification, whatever the bioburden at the dirty clean room, let's say, is.
Thank you, Michelle, for that question. We have a question from Prerna, which is, what's the way to remove mold from cold room? Spraying with more disinfectant may end up creating more wet surfaces, which is hard to dry because of low temperature.
So generally mold come from the, I always say, come from the exterior. And I think Jim Polarine and even Richard will agree with that. So the first things to look is the root cause of where it is coming from. The second thing, you're right, that putting a disinfectant, maybe it's more appropriate to use a sporicide for such type of cold room. And if possible, the best way is to have this this cold room being declassifier where you can decrease the temperature, increase the temperature, and perform a full cleaning and then restart your clean room, especially if the mold starts to pop up everywhere. Why? Is because your wet contact time of a disinfecting agent, especially of a sporicide or a disinfectant, will be multiplied by even 4 or 5 times. So you have to demonstrate that either through a coupon study at a cold room, if you have a lots of cold room at the temperature that you are working, what is the adequate wet contact time, or perform if the excursion is not very important or very recurrent, there is a possibility of using a detergent to remove and then use a sporicide to make sure you have a kill and make sure you wait the adequate wet contact time.
Thank you. And Mark Quo asks the question, what's the way to remove mold from cold room? Oh, sorry, that's a similar question. And he's put, we at Sanofi would close down the cold room and clean it down with NaClO and H2O2.
Yeah, so NaClO is like bleach, and H2O2, I guess, is a mix of those both, because H2O2 or VHP, NaClO is can be a sporicidal agent at a certain concentration, and can, as a sporicide, will help in removing the mold. However, you want to make sure your surface is compatible with NaClO. Another thing with NaClO, bleach, is that you want to make sure you have removed any organic residue that may alter the efficacy of this disinfecting agent. If H2O2 is used as a VHP, then that should be okay. If it is used at 6 or 3%, then I'm not sure it will be efficacious at a normal ambient temperature wet contact time against a spore former mold or even a vegetative mold.
Thank you. We have one question from Pierre Michel about the stopper bowl. Why would you consider it has not to be disassembled?
No.
It could be cleaned and autoclaved. Does Annex 1 not push doing so?
Yeah, no, okay. Maybe I'm misunderstand, or maybe I wasn't very clear. The way to do it is to disassemble it if it is possible to disassemble it, clean it, autoclave it, and that's the way. You're right. The Annex 1 is pushing to it, and it's not something new. In some cases, some of those stopper bowl it is impossible to disassemble it. Let's say like that. And then the only way is to, like I exposed in the presentation, is to be able to perform a cleaning of it and to decontaminate it as a surface that is fixed in the wraps or in the isolator. Thank you for this question, because it clarifies if there is any other type of misunderstanding or any other person that may have understood that. And I'm sorry if that's the case.
Thanks, Walid. Thanks, Pierre Michel. I think we've answered all the questions, and apologies to Neo, who is Walid and Richard's colleague, he also answered the mold question earlier as well. We have 4 more minutes, so this is your last chance to pose any questions, or please feel free to unmute yourselves. We also have some questions for you under the evaluation, so we'd appreciate if you would click the link that we sent to you as well. Prerna asks, what are the key points when performing cleaning validation for column containing resin?
Yes. Richard, do you want to go or I go?
Yeah, I just answer first. So this one is basically on the product contact cleaning.
Yes.
It's not related to what we presented today, but
exactly.
This one is basically cleaning. You need to do the unpacking of the resin first before you perform the cleaning using the detergent. And because you will need to basically check that the detergent is compatible with your resin. So typically you will need to unpack first, then you perform the cleaning on the column.
Exactly.
Mm-hmm.
So you will have to clean the resin if there is, but with a solution that you know is sodium hydroxide or a commodity like we call. But then after depacking the column, then there is a possibility to clean the column itself. Again, you want to make sure, because resin is very expensive, you want to make sure that any product that you are using is compatible, like Richard say. Otherwise, you would just ruin your resin. I remember when I worked in the industry, it was a time where we tried to dedicate those resin, but then there are some regulation that now allows a certain multi-product, but I'm not sure. I need to look and find out. We may come back to you.
Yeah, you can contact us separately to discuss in more detail, on your cleaning requirement.
Thank you, gentlemen. And on that point, we will be sending an email to everyone that's registered to your email address with the links to this recording, as well as to the handouts, and also with the contacts for Walid and Richard for follow-up. So if you have any direct questions, you may ask then. We also have the link to the evaluation, and we would appreciate that. So this is, we have one last question, I think. Or no, I think that all the questions have been answered. So with 2 minutes left to go, if anyone wants to unmute themselves and get in there before we finish for the evening. Otherwise, you are most welcome to pose your questions to our experts directly. And we want to thank everybody. Mark says he's hungry, it's dinner time here. So unless there are any further questions, we want to thank everybody for joining us for this session, and to our experts, Walid and Richard, for sharing their time and expertise with us. And without further ado, we'd like to finish off this session and to wish everyone a lovely evening, and to Walid a great day ahead. I know it's the start of your work day and the end of ours. And we look forward to seeing you all to join us for STERIS's next session, which will be in September. But if you have any questions, please do pose and let us know. So thank you all, and have a great evening.
Okay, thank you.
Bye-bye, everyone.
Bye-bye.